Hi, I am Chirantan Rawal. Today we will have a talk on the introduction to bacteria. That is part 1. The purpose of this chapter is to describe the structural organization of the prokaryotic cell, that is, a bacteria. The bacterial cell represents the simplest of all cellular organisms when seen under the microscope. Bacteria is a prokaryotic structure. The singular for this word is bacterium. They do not have true nucleus. They have one chromosome of double-stranded DNA in a circular form. They reproduce by binary fission. They do not have any intracellular organelles. They belong to kingdom Monera, which is very diverse group. There are some photosynthetic bacteria which can do photosynthesis. They do not have a chloroplast, but their photosynthetic pigments are their cell membranes. It means the photosynthetic pigments are lying in the cell membranes. These cells are called blue-green algae or a cyanobacteria. They are the most common and omnipresent microorganism on the earth. So student, tell me, what is a cell theory? Anybody? Okay, I will tell you. The cell is the structural and functional unit of life. All organisms are made up of cells. New cells arise from the pre-existing cells. So these are the statements of the cell theory that explain us about the basic structure of life that includes the cell. What is a structure of a bacterial cell? Do you know? Anybody? Okay, I will tell you. A variety of structures are found in prokaryotic cells. If you go through the typical image of the bacteria, you will find a cell membrane. In some cases, you will find the capsules. They have the cell wall. They have a circular chromosome. They do not have a nucleus. Even they do not have a membrane-bound organelles like chloroplast, mitochondria. Such organelles are absent in the bacteria, and that's why they are included in the prokaryotic group. When you go through the typical image of the bacteria, you will find a plasma membrane, cytoplasm, cell wall, DNA, ribosomes, flagella, pili. But all these structures are not found in all genus. Gram-positive and gram-negative bacteria differs in their cell wall content. All bacterial cells were surrounded by a cell wall except mycoplasm. After cell wall, there is a periplasmic space, beneath which a plasma membrane lies. Bacteria does not contain the intracellular organelles, so their internal structure is very simple. Genetic material consists of the circular chromosome, which lies in the nucleoid region. Ribosomes and the inclusion bodies are scattered in the cytoplasm. You must know that the ribosomes are the protein building machinery. They synthesize the protein from the RNA. Many cells have flagella for locomotion. Pili is also observed for sexual reproduction. Some bacteria also contains capsules surrounding their cell wall. Now, let's try to understand more about the morphology of bacteria. The shape. Generally, 4 representative shapes of bacteria are observed. That is coccus, or you can say cocci. These bacteria are spherical in shape. Coccus is a singular and cocci is a plural. The second, the bacillus or bacilli. They are rod-shaped. Third, spirillum or spirilla. They are spiral-shaped. And the fourth one is the comma-shaped cells, that is the vibrio. You must be knowing the vibrio cholera, that is causing the cholera in the human being. They are comma-shaped cells. Apart from these shapes, some typical shapes of bacteria are also observed. But these shapes are restricted to few species only. The square-shaped bacteria is reported in the year 1980 by A.E. wax bee. Star-shaped bacteria is also reported that belongs to the Stella genus. Actinomycetes characteristically form long multinucleate filament structure. Actually, we believe that the actinomycetes are connective link between the fungi and bacteria. That's why their shape resembles the fungal growth. Actually, we believe that the actinomycetes are the connective link between the fungi and bacteria, and that's why their shape is resembling the fungal growth. Apart from this, a pleomorphism is also observed. That is, some bacteria does not have a particular shape. They are variable in shape and lack a single characteristic form, and that's why they are called apleomorphic. Their shape is not perfect. Their shape is changing from generation after generation. Like that is in the case of amoeba. That is also apleomorphic. Their shape we cannot determine. The similar pattern is also observed in some bacteria. Now, let's try to understand the pattern of cell arrangement. I repeat, the cell arrangement. The bacterial cell may be categorized according to arrangement or cell grouping. Every bacterial species have their characteristic arrangement pattern. Variety of arrangement pattern is observed in cocci. Most common arrangement patterns are single, a diplococcus, that is a pair of cells, streptococcus, that is a chain of cells, tetrads, that is a 4 cocci in a box-like arrangement, sarcinae, that is a cuboidal packets, staphylococci, that is a cluster of cocci. Then question must arise in your mind. What is a size of bacteria? The bacteria range in a size from about 0.2 to 5 micrometer. Smallest bacteria are mycoplasma, which are about the same size as the largest viruses like pox viruses. Cocci measures anywhere from 0.5 to 3 micrometer in diameter. Bacilli range from 0.2 to 2 micrometer in diameter and from 0.5 to 250 micrometer in length. E. coli, that is Escherichia coli, has the size of 1.1 to 1.5 micrometer and they are 2 to 6 micrometer long. The next is the habitats of bacteria, means where they are living, where they are observed. They are found in the bottom of the deepest oceans, as well as they are also found at the peak of the mountain. They are found inside the body of animal and even in the frozen rocks and even on the ice of Antarctica. Means they are omnipresent. One feature that has enabled them to spread so far and last so long is their ability to go dormant for an extended period. They are found even in the hot springs, frozen soils, acid-drained mines, and the saline habitats. If you go through the habitats of the archaebacteria, you will find that they are observed in the variety of habitats, in the extremes. And they are also known as extremophiles, particularly the archaebacteria. Next, we will learn about the reproduction in bacteria. Bacteria reproduce very rapidly. You will not believe that the E coli. Next is the reproduction in bacteria. Bacteria reproduces very rapidly. You may not believe that, but E. coli can divide within 20 minutes. Bacteria reproduce by 2 basic methods. They are categorized as asexual reproduction and sexual reproduction. First, we will try to learn about the asexual reproduction. Asexual reproduction involves only one parent. The offsprings are exact copy of the parent. Bacteria splits into 2 cells using binary fission. Each daughter cell receives the same genetic material like parent cell. The next is the sexual reproduction. It requires the 2 parent cells. It involves the physical contact between the 2 cells. It occurs with the help of the conjugation tube. Here, progeny cell have a mixture of the parent cell's traits. It occurs by the process of conjugation. Now, let's try to understand the various structures that are found in this bacterial cell. We will divide these structures as a structure external to the cell wall and the structural internal to the cell wall. In this talk, we will try to understand the structures that are external to the cell wall, while in the next talk I will discuss about the structures internal to the cell wall. So, what are the structures that are found external to the cell walls? That is, a flagella, pili, fimbriae, prosthesi, stalks, spines, spirae, capsules, slime layers, and seeds. We will learn one by one. The first, I will discuss the flagella. Majority of the motile bacteria moves with the help of flagella. It is a thread-like appendages extending outward from the plasma membrane and cell wall. Size of the flagella is 15 to 20 micrometer long. The detailed structure of a flagellum can only be seen through electron microscope. What is flagellation? Or what is a flagellar arrangement? There are different arrangements of flagella found on a bacterial cell. Different patterns of flagellation are monotrichous, amphitrichous, lophotrichous, and peritrichous. In monotrichous, one flagella is there that is located at one end. For example, in Pseudomonas species, monotrichous flagella is observed. Amphitrichous means two flagella located at each wall. For example, in the Spirulu. Lophotrichous, that is a cluster of flagella at one end. Example, Aquaspirillum. Peritrichous, that is many flagella spreads evenly on wall surface. That is found in the Proteus species. Ultrastructure of a flagella. It consists of three parts: filament, basal body, and hook. Filament is the longest portion extended from the cell surface. Basal body is the part which is embedded in the cell body, while the hook is the curved part which connects the filament to the basal body. Basal body is most complex structure of flagella. In E. coli, basal body consists of 4 rings, that is L, P, S, M rings, while gram-positive bacteria have only 2 basal rings. And what is the chemical composition of a flagella? It is made up of fibers. These protein fibers are made up of flagellin protein. Molecular weight of the flagellin protein is in the range of 20 kd to 60 kd. The kd means kilodalton. The flagellin protein of a different bacterial species is not identical. How flagella helps in the movement? That is a mechanism of flagellar movement. Bacteria moves when this flagellar helix rotates. Direction of flagellar rotation determines the nature of bacterial movement. The monotrichous polar flagella rotates counterclockwise during normal forward movement. Monotrichous bacteria stop and tumble randomly by reversing the direction of flagellar rotation. Peritrichous flagella forms bundle of the flagella and moves in the similar way. It is believed that flagella moves due to interaction of the rings present in the basal body. There are different kinds of motilities observed, like swimming motility, gliding motility. In the swimming motility, the examples are spirochetes. Spirochetes moves due to the axial filament present in between the perisplasmic space. It is considered as a flagella and that is present inside the body, and that's why it is also known as endoflagellate. You must be knowing the periplasmic space. That is a space present between the cell wall and the cell membrane. The next is the gliding motility. This movement occurs without flagella. Gliding bacteria are able to move on a solid surfaces. Actually, they glides, and that's why it is known as gliding motility. The next structure outside the cell wall is the fimbriae and pili. A fimbriae is used to denote the non-flagellar appendages other than those involved in sexual conjugation. Pili refers to the appendages that are involved in the transfer of nucleic acid during a sexual process between the two bacteria, that is conjugation. They are found mostly on gram-negative bacteria, particularly on Enterobacteriaceae family. The next structure, prosthesis. It is an extension of the cell wall and cytoplasmic membrane. They are observed in bacteria which are found in the freshwater and marine environment. For example, Colobacter stella. It increases the surfaces of the cell and which helps them the absorption in the nutrition. The next is the stalk. Stalks are the non-living tubular extension that are excreted by the membrane of the genus Gallionella and Planktomyces. It aids to attachment of the cell to the surfaces. The next is spines. Some marine gram-negative bacteria exhibits this structure. It is a rigid protein appendages. It protects bacteria against ingestion by larger microorganisms. The next structure is spirae. In nitrogen-fixing bacteria like Azospirillum lipoferum, which produces several spiral structures called spirae. It is a single coiled thread-like structure. Its function is not known to us. The next structure is capsule and slime layers. Bacterial cell exude extracellular substance in the form of capsule or a slime layer. A slime layer is loosely connected with the bacterial cell and can be simply removed, whereas a capsule is attached firmly to the bacterial cell. Capsule has a specific margins. Capsule can be seen under a light microscope using a capsule staining India ink. Capsule does not stain by the stain and appear as a clear halos surrounding the bacterial cells. Capsules are made up of simple sugars, that is the polysaccharides. Most capsules are hydrophilic and may help the bacterium to evade the dehydration by preventing the water loss. Capsule can protect a bacteria from phagocytosis. It may be due to more slippery nature of the capsule, which helps the bacterium to form engulfment by WBC. Capsules are also related to the pathogenicity of the bacteria. For example, Streptococcus pneumoniae. The S. pneumoniae mutants which are not able to synthesize the capsule, they do not cause disease. Such association of pathogenicity and capsule is also found in some other bacteria. It is also associated with biofilm formation. The next structure is sheath. Some species of bacteria, particularly those from freshwater and marine environments, forms a chains or a trichomes that are enclosed by a hollow tube called sheaths. It may be sometimes becomes impregnated with ferric or manganese hydroxide, which strengthen them. This is all about the structures that are found external to the cell wall. In my next talk, we will discuss about the structures that are found internal to the cell wall and the cell wall itself. Okay, thank you very much.